Assalamualaikum warahmatullahi wabarakatuh, and a very good day everyone. So we're coming to the third lesson video on fertilization and fetal development. So this is 9.3 from chapter 9. We first will begin our lecture by focusing on fetal development. That means we are going to focus on what happened after fertilization are successful. Okay, in this lesson video, by the end of it, you should be able to first define embryogenesis. So this is one of the process that we call overall event that occur during fetal development. Second, to state the development stages from which the zygote will develop to form a structure called morula, blastocyst, and gastrula through a series of cleavage. Okay. So in this image, it serves to help illustrate the flow of the process, and you could imagine what happened as an overview to the structure of the zygote. Okay, so for fetal development, it all began from the structure in the female reproductive organ, which is the ovary. Okay, so this is one of the ovary. Okay, as we all know, inside ovary, so that's where the development of the gamete occurs. Okay, as a result of that, so we will produce a secondary oocyte which will leave the ovary through the ovulation. Okay, every menstrual cycle. Through ovulation, once this oocyte left the ovary, so it will enter the fallopian tube, okay, in which it will continue to the next stage called the fertilization. Okay, so general overview of fertilization is secondary oocyte made up with the sperm. Okay, and once these are successful, so we are going to produce a zygote. Okay, and zygote will further develop along the way until they are capable of undergoing the next stage, which is called the implantation process. Okay, so what happens is that throughout this development process, okay, zygote will undergo series of changes to its structure. Okay, and within a week of development, implantation allow the structure to be able to be embedded within the endometrium, which is the wall of the uterus here. So and this allow the further development of next structure called the embryo. Okay, right. So what actually happened between zygote to the implanted embryo? Okay, so as you see in this illustration, okay, can you spot the difference to the cells that are present in each of the structure? Yes, you are right. So what we can see is that there is changes in terms of number and the sizes of the cells. Okay, so what happened first to this, okay, is a series of changes to the number. So as we know, number of cells are added to original structure by division. Okay, so here the flow shows the number of cells being added from 2 until eventually about 100 of them in a structure called morula. Okay, and then from this ball of cells, okay, so it further develop in the next process called gastrulation. So the focus of gastrulation is layers. Okay, layering. And eventually there will be new relation which we won't take into account, or we are not going to discuss any further in this syllabus. However, basically it's focusing on the development of the neurons, okay, and obviously the nervous system, and a newborn completely formed at the end of the process. Okay, so once a good structure of embryo develop, so within each before they actually finally form a good complete newborn or fetus, So they are going to undergo formation of organ process. So basically, as the name suggested, so each of these tissues and cells will specialize to become a specific structure which conducts specific function, known as the organ. Okay. So for formation of embryo and its development, so this process is called embryogenesis, as the name suggested. So genesis always refer to the synthesis or the formation, okay, and the embryo, of course. Okay, so the human development can be summarized to form embryo through first gametogenesis. Okay, so from the word gamete, and the genesis means form. Okay, forming gamete, followed by formation of successful gamete, of course fertilized. So this is the fusion, as I shown earlier. Okay, and we discussed these fertilization stages in detail on other lesson. Okay, and then the human will further through cleavage. So this is dividing process, which we focus on the addition of the number of cells. And after cleavage, there will be gastrulation, the layering, okay, forming tissue layers, and finally organogenesis, synthesized organ, okay, form organ. And from the organ developed, so we will continue growing. Through the growth, the tissue that develop within each of these organ will undergo specialization. So that means they have specific function to conduct. Okay. So what is the first stage, which is the cleavage? Okay. So cleavage, the focus is division. Okay. But in terms of definition, so we focus on a series of rapid cell division during early development. So there are a sequence of this cell division. They are rapid, so they take a small amount of time. So as I shown earlier in the illustration, so between this series of cleavage until implantation, so it take around 1 week only. Okay, so that's why we refer to as a rapid division. And since division occur, so each of these cells divide from 2 to 4, 4 to 8, 8 to 16, and so on until they form these huge balls. Okay, so that is cleavage, right? And then showing here, okay, so successful cleavage will eventually form a structure called the blastocyst. Okay, so blastocyst is shown here when the ball of the cells have a space form. Okay, so within this space there will be liquid, and then it help enlarging the size of the embryo. Okay, right. Okay, and then obviously next stage is through I forgot. So this is called morula, and at the end we have the blastocyst. Okay, and then after implantation, so shown here. Okay, so basically the blastocyst will be embedded into the wall of the endometrium through implantation. So we have this few structure form, which is the layering of this tissue allow formation of epiblast, hypoblast, and trophoblast. So trophoblast is referred to the outermost layer tissue, hypo in the middle here, and this would be the epiblast. Okay, and then from this hypo and epiblast, they further develop. So what basically happen is the tissue layers are like migrating and rearrange themselves, so they focus on a specific area on the embryo. Okay, and the important parts of this rearrangement is so that the embedded embryo capable of obtaining nutrient, and they can form a structure like chorion and yolk sac. Okay, so to provide the nutrients to the developing embryo. Right, and then stage 2 of the development, as mentioned earlier, are gastrulation. Okay, so from gastrula, so we will end up with a gastrula. So as I mentioned, gastrulation is basically layering. Okay, so what layer they form is an what we call as embryonic layer. We call it germ layer. Okay, so that's the basic tissue layers formed within the embryo, which will determine the fate of each tissue to form a specific structure later in the next development. So gastrula for human, basically the arrangement will form a 3-layered tissue, meso, ecto and endo. Okay, ecto is the outer layer here, the blue colored. Okay, and then endoderm, okay, would be the innermost layer, the green color, and the mesoderm, the layer in between ecto and endo. So this region over here. Okay, so this is gastrula. Okay, so 3-layered embryo. So the 3-layered embryo are formation of 3 germ layers that determine the fate of each tissue in the next stage, which is the organogenesis. Okay, so this is just to summarize. From zygote to the blastula undergone formation of morula first. So basically they rapidly divide, and then from blastula they form this space which contain liquid in it. And then tissue from the blastula rearrange to form 3 germ layers. Okay, but here it only shown endoderm and ectoderm. So this is different species. So depend on the species, so the germ layer will be different. So for human, we have 3 layers. Okay, and then the final part of the chapter or the fetal development stage is focusing on organogenesis. So I mentioned earlier on that each of these tissue layer from gastrula will be determined to be fated to form specific structures. So that's the organogenesis, formation of organ. Okay, what is organogenesis? That's the first learning outcome, and the second is what are the organ forms. So basically, what organogenesis, define it, and then determine which organ form from which germ layer. Okay, right. So organogenesis, is the region of 3 embryonic germ layer develop into rudiments of organs. Simple. So from the germ layer, the 3 layers, ectoderm, mesoderm, and endoderm, each of them becomes organ. Okay, so what organ do they develop? Okay, so we start off with, so for this image, so this to illustrate the regions where ectoderm, mesoderm, endoderm originally were formed, and then finally for the dorsal ectoderm. So all this term is to show whether it's the frontal or the back part of the ectoderm. Okay, so ectoderm, which is the blue colored, on the dorsal region, we will form a neural tube, hollow neural tube, which is eventually form the central nervous system, which is the brain and spinal cord. So this is the dorsal part. Okay, and then you have the notochord formed from the dorsal mesoderm. Okay, mesoderm is the pink colored one. So dorsal means the frontal. Okay, so here, so that will be the notochord formation. So this is only found usually during the embryonic development. And then finally mesoderm layer, so all this pinkish structure, so they grow into we call this as somites. Okay, but at the end of this development, so they will form the vertebrae and the muscles of the human. Okay, right, and major derivative of 3 embryonic germ layer develop into specific structure. For example, ectoderm, because they are the outer layer, so they form outer part of the body. It could be epidermis of skin, the nervous and sensory system. So we know that the layer, the first layer that we found under the skin is the nervous system, so our sensory system. And then it also helps in the formation of pituitary gland in our brain, jaws and teeth as well, coming out from ectoderm. And then we have mesoderm layer, which is the middle layer, so that's responsible for muscle and skeletal development. the reproductive system, and then adrenal cortex. So this is on top of the kidney. Okay, and for endoderm, which is the innermost layer of the germ layer in embryo, so they further finally will develop into the epithelial lining of digestive tract. So you can remember one of these example for each of the layer to help you determine the different types. Okay, so that's it for the lecture. Thank you.